Cyanobacteria.
It's a bacterium, good. And it belongs to this cool group of bacteria called Cyanobacteria. In the old days, they called Cyanobacteria blue-green algae, but that's such a bad name because algae are eukaryotes, right? Are Cyanobacteria eukaryotes? Do they have a nucleus? No. Okay, so we don't want to use the term blue-green algae. Some microbooks today still use that term, like egads. Okay, they're prokaryotes. They belong to this very cool group called Cyanobacteria, and the genus name is Nostoc. Okay, and because it's part of the scientific name, I always what? If I'm writing on the board, I underline it, right? Okay, so let's talk about it. So when you're looking at your Nostoc, you'll see these lovely chains of cells. Each ball is one cell. So these guys are what? Unicellular. They're unicellular. They're unicellular. And when they divide in two, often they remain attached to one another. So you'll see these beautiful chains. Oops, and there's the green. I'm so sorry. I said I wasn't going to use green. Okay. So we got one cell. They're unicellular. And then they'll form these beautiful chains that you'll see. Now, on scanning lens, the little chains, they look like little worms, okay? So don't stop there. Move up low power on high dry, and on high dry you'll see those little worms are actually these chains of cells. Okay. Now, what is cool about cyanobacteria? What's cool about Nostoc? They carry out two really important processes. And this is more information, you guys, it would be fair game to ask you on the lab practical, okay? So these cyanobacteria, they carry out two incredible processes. They carry out oxygenic photosynthesis. And if we were to write a little summary reaction for oxygenic photosynthesis, they can take inorganic carbon dioxide from air, and in the presence of water, they can convert light energy with the help of a beautiful photosynthetic pigment called chlorophyll A. They can convert the carbon dioxide and water into organic molecules, such as glucose. Yeah, isn't that wild? And oxygen, that's right. And I'll put an arrow pointing up with molecular oxygen to show it's a gas. Okay, this is phenomenal. These organisms can take light energy and convert it into chemical energy. That is amazing. Okay. So we rely on organisms such as cyanobacteria. We call these organisms that can synthesize their own organic molecules from inorganic precursors using what we call photoautotrophs. We rely on them to make these organic molecules that we then use as a source of carbon and energy. Yeah, so they're really food makers for us. And as if that wasn't incredible enough, cyanobacteria have another incredible process called nitrogen fixation. In chapter 7, prokaryotic ecology, which we'll do before the lab practical, we'll explore nitrogen fixation in more detail. But just to give you a preview, you guys, the cyanobacteria in the process of nitrogen fixation, they take molecular nitrogen from air and they convert it into ammonia. And you're like, well, so what? Who cares? Well, it turns out that cellular organisms, we cannot use molecular nitrogen as a source of nitrogen to make amino acids, to make nucleic acids. We have to have it converted into ammonia before we can use it to make organic molecules. Very few organisms can carry out this process of converting the unusable molecular nitrogen to usable ammonia. The cyanobacteria can carry out this process. So again, it's like, oh my gosh, I should be bowing down before these. Nostoc, oxygenic photosynthesis, making new food. Nitrogen fixation, converting unusable nitrogen to a form that we can use to make amino acids, to make nucleotides or nucleic acids. This little tiny baby cells, you guys, it's just, wow, I wish I could just do part of what they can do, okay? All right, so I would recommend that you try drawing these guys, and remember, on the lab practical, you're gonna sit down at a station, we'll have a microscope there, of course we're gonna cover the label, you're gonna have to look through the scope and know, oh, I know, that's Nostoc. I know how to classify it, I know these two incredible processes it carries out, and there's one more thing, you guys, that I want you to know. According to the endosymbiotic theory, primitive cyanobacteria evolved into eukaryotic chloroplasts. Okay, so this is something we'll be discussing in lecture as well. And again, I'm using that doggone green. Okay, so we're just going to show a line that, that leads from cyanobacteria over here to the main eukarya. And that line is to indicate that, according to the endosymbiotic theory, eukaryotic chloroplasts evolved from primitive cyanobacteria. There is amazing proof that supports that theory. One is chloroplasts inside algae and plants, like the Elodea leaves that you're going to look at today. Those chloroplasts, which we think evolved from cyanobacteria, they have their own circular DNA that looks just like a bacterial chromosome. The ribosomes inside chloroplasts look just like bacterial ribosomes. We're going to find out in lecture next week that us eukaryotes, our ribosomes in our cytoplasm are big ribosomes. In contrast, bacteria, the ribosomes in their cytoplasm are small ribosomes. Chloroplasts have their own ribosomes. Do you think they're big or little? They're little, just like a bacterial ribosome, right? So this is all really powerful evidence that supports this hypothesis that eukaryotic chloroplasts that we see in plants like Elodea and in algae actually evolved from primitive cyanobacteria. So that's a connection here, you guys, I want you to make, okay, between these two organisms. Yeah, yeah.
So you said the ribosomes resemble, and what else resemble?
The chloroplasts have their own circular DNA, which looks just like a bacterial chromosome. Yeah, just amazing. Okay, so talking about plants, then let's talk about the other organism that you guys are going to be looking at today. And over here, we'll put photomicrographs diagrams that Professor Holland has downloaded. Heads up, you guys, these often appear on the lab practical, so don't ignore them. So one of the dynamite photos, or microphotographs Professor Holland downloaded was of Elodea, the Elodea leaf. These little boxes are individual Elodea cells. Okay, so as a plant, they have a strong cell wall. What is the polysaccharide in the cell wall that helps to strengthen these guys? We talked about in lecture today. Cellulose, good. Okay, now what we want you to really focus on in your wet mounts today are these really cool little circular structures inside the Elodea leaf. What's the name of those little green organelles? Those are the chloroplasts, okay? In eukaryotes, chlorophyll A, the photosynthetic pigment that permits oxygenic photosynthesis, chlorophyll A is found inside chloroplasts. Okay, what if I asked you, according to the endosymbiotic theory, how did chloroplasts evolve? Where did they come from? Cyanobacteria, exactly. And again, you guys, if we could look inside those chloroplasts, we'd find a bacterial-like circular DNA molecule, and we'd find ribosomes that look just like bacterial ribosomes. Now, the chloroplasts themselves are not motile, but if you're patient, you will see the chloroplasts moving around the periphery of the cell in a process called cytoplasmic streaming. The center of the Elodea leaf, it has a fluid-filled vacuole called a central vacuole. Okay, so